Hi dear students, welcome to Ardentmds. So this is JVE just before exam. The motto of the JVE is more important points in a less time. Without much wasting time, we go for a topic, molecular diagnostic techniques in COVID-19. There are three important tests done for the COVID-19. One is a direct test, indirect test and supplementary test. In case of a direct test, the most gold standard test till now is RTPCR. Reverse transcriptase polymerase chain reaction. Apart from this, we can go for antigen detection test, gene expert test and a TruNAT test. And gene expert test and a TruNAT test both are previously used for tuberculosis. Now it is used for COVID-19 also. Antibody detection test will come under indirect, chest X-ray and high resolution CT and interleukin levels will come under supplementary test. Let's start with the RT-PCR. We have to collect two swabs, one from the nasopharyngeal, one from the oropharyngeal, according to the criteria. So how to collect the nasopharyngeal swabs? What are the step-by-step procedure? So remove the swab from the pack and ask the patient to tilt the head slightly and insert the swab along the nasal septum up to you feel a resistance. That means you reach the nasopharyngeal area. Gently rub and roll the swab, collect the secretions and remove the swab slowly while rotating. So once you collected the swab and you have to cut down the extra portion and insert into the viral transport medium at the temperature of 2 to 8 degrees Celsius and transport to the laboratory. How about the oropharyngeal swab? Insert the swab into the posterior pharynx and tonsillar areas. So collecting the secretions from the posterior oropharynx. So again same as a nasopharyngeal swab, cut down the extra thing and put it in the viral transport medium. At the temperature of 2 to 8 degrees Celsius, it is sent to the laboratory. Right. So what about this viral transport medium? What is the composition? So it contains 10 gram of viral infusion broth with 2 gram bovine albumin, sterile distilled water, and most importantly, it has antibacterial and antifungal agents like gentamicin and amphotericin to avoid the contamination of bacteria and fungi. Finally, it's sterilized by the filtration process. Right. So once the swab is collected and put it in the transport medium and send it to the laboratory person, in lab they go for a procedure called as RT-PCR, reverse transcriptase polymerase chain reaction. So here usually in a PCR the DNA is amplified. Usually in a PCR the DNA is amplified. But here we collected SARS-CoV-2 which has RNA. So this RNA have to be converted into DNA. So usually what is called as transcription is converting the DNA into RNA. But here we do just opposite procedure, converting the RNA into DNA. So converting the RNA to DNA by done called as reverse transcriptase. So the enzyme reverse transcriptase is added to convert the RNA into DNA. So this DNA along with the primer and nucleotide, Taq polymerase and buffering solutions. So all these are which joins together and inserted into the PCR tube. So this PCR tube kept it in a thermal cycler. So as the name says thermal cycler, it produces cycle of high temperature and low temperature and it go for a process called as denaturation, annealing, extension procedure and finally we get the results. What are the gene targets for RT-PCR? You have two type of genes. One is a semi-confirmatory genes and confirmatory genes. What about semi-confirmatory genes? So yesterday in the introduction part we studied about SMEN or the proteins, right? Just except this membrane protein, spike protein, envelope protein and nucleoprotein. So these are the gene targets for the RT-PCR. And coming to the confirmatory genes, ORF1A that is open reading frame 1A and RDRP that is RNA dependent RNA polymerase are the confirmatory gene for the RT-PCR test. At the end of the test, you will get a results like this. Envelope gene is positive, but if the RDRP gene is negative, that means the patient does not have a COVID-19 positive case. Right? In case of envelope gene is positive and RDRP is also positive, then you can consider about COVID-19 positive. And E gene is positive and open reading frame also positive, then the patient is considered as COVID-19. So how to interpret the results? All this which is coming in a fluorescent flash like this, so which gives a ratio of the control and experimental and we will get the final results like this and computer which gives a flash like this. So based on this we will get a results. Right, coming to the comparison of RT-PCR and TruNAT. RT-PCR which has both high sensitivity and specificity than TruNAT, and if you see this, it has a specificity of 95% and sensitivity of 70 to 80%. RT-PCR can detect even 10 copies per ml, but TruNAT which detects 486 copies per ml, and it takes time 4 to 6 hours, but TruNAT which requires just 60 to 90 minutes, which is the more cost effective. RT-PCR is more cost effective than TruNAT. By coming to the cycle threshold value which gives an idea about relative viral RNA estimation. Usually the CT value is just indirectly proportional to the viral RNA levels, mean more CT value the patient has less viral RNA. So example, if the CT value is 30, that means patient does not have able to spread the disease to the others. Coming to the immunochromatography test which detects both antigen or antibody. So here just collect the swab, put it in a virus lysis buffer, take 1 to 2 drops and put it in the cord and you will get the results. It is like act like a pregnancy test, home pregnancy test. So the uses are antigen test and antibody test. Antigen test which is used for diagnosing the particular infection, but antibody test which gives about idea of seroprevalence in a particular area. So these are the various points which you have to know related to the molecular diagnostic techniques for the COVID-19. Now it is your turn. So you have to answer for the Ardent golden points related to the COVID-19. Gold standard laboratory diagnostic test for COVID-19 is RT-PCR. Right. Antibacterial and antifungal agents added in viral transport medium is gentamicin and amphotericin. Nodal laboratory for COVID-19 in India. Nodal laboratory for COVID-19 in India is NIV, that is National Institute of Virology. They are in Pune. Genes targeted in RT-PCR is semi-confirmatory and confirmatory. Semi-confirmatory are spike protein, envelope protein, nucleoprotein. So those genes which controls this protein. And confirmatory genes are RDRP, that is RNA dependent RNA polymerase, ORF only, open reading frame. Cyclic threshold value is directly proportional to the viral load. No, it is a false statement. Cyclic threshold and viral load is indirectly proportional. Which test has high sensitivity for the COVID-19? Yes, RT-PCR has the high sensitivity. Type of RNA which is detected and highly sensitive in RT-PCR is mRNA. And what is the full form of POCT? That is point of care testing. So next is about Ardent flash. All the important points till now what we studied, which is given in Ardent flash here. So just go through this laboratory diagnosis. What is the composition of viral transport medium? Results of RT-PCR, how to interpret, and about cyclic threshold value, comparison of RT-PCR and TruNAT, and immunochromatography test. Just take a pass and go through all the important points related to the molecular diagnostic test for COVID-19. So coming to the important questions related to the molecular diagnostic testing, point of care testing is for the detection of both IgG and IgM. So answer is D. What is point of care testing? Instead of sending all the samples to the third party laboratory or the central hospitals, so there are some local laboratories which just present to the near to the patients. So this is called as point of care testing. So which reduces the workload to the central hospitals and the central laboratories. Least recommended sample for COVID-19 is bronchoscopy, which has risk of high aerosol. All the following are calculated tools for the predict critical illness in COVID-19. So LDH and elevated neutrophil lymphocyte ratio, that means the patient is having critical illness. Right. Indirect bilirubin does not anywhere related to the critical illness of COVID-19 patients. So B and C, which gives an idea about critical illness, and decreasing the LDH and neutrophil lymphocyte ratio, that means the patient is improving. So these are the biological markers for the prognosis. In case of asymptomatic phase, mild lymphopenia is seen. Here one of the very important thing. So usually lymphocyte is decreased in case of the initial stage and mild to moderate stage. So again in a mild to moderate, patient is having lymphopenia, mild increase in ferritin and D dimer level. In case of a severe cases, D dimer is elevated, ferritin is elevated, and the patient is having abnormal blood gas analysis. Even interleukin 6, C reactive protein, all these are elevated in case of severe. In critical case, elevation of inflammatory markers are seen, and the patient also has increased renal function markers. In case of a serological examination, if the PCR is positive but IgM and IgG is negative, that means the patient is still in the window period. In case of a serological examination, if the PCR and IgM is positive and IgG is negative, so what does that mean? The patient is in the early stage of infection. So here you have to know the concept. So there are two immunoglobulins which you have to take it point, IgG and IgM. IgM usually seen in acute infection. IgG is chronic infection. In case of a early phase or acute infection, IgM you can expect it to present, but in case of a chronic infection, IgG is present, right? And IgG which appear later part of the disease, disappear also later. So based on this concept which you can able to answer. In case of a serological examination, PCR, IgG and IgM all are positive. If all are positive, that means the patient is in a active phase of infection, next stage. But in case if the PCR is negative but IgM is positive, that means what? Patient is either in the early stage of infection or it may a false negative. So both A and B. Right. So this is one more question. PCR and IgM is negative but IgG is positive. So because IgG is positive, then you can say it is a post recovered infection. The next type of question related to PCR is past infection with COVID-19 is detected by RT-PCR. Is it true or false? Past infection with COVID-19 does not able to detected by RT-PCR. So this is a false statement. So these are the various things which you have to know related to the PCR test about IgM, which is seen in early phase, and in the middle phase IgG and IgM, RT-PCR, all these are positive. But in case of a recovered patient, RT-PCR is negative. Right. Coming to the laboratory investigation in COVID-19, elevated levels of all the following are seen except lymphocytes. Usually we studied lymphocytes are decreased. All the following categories will be tested for COVID-19 except, right, all the symptomatic individual, the patient who had a history of international travel in the last 14 days, yes, the patient should go for COVID test. All symptomatic healthcare workers, yes, they have to go for COVID test. Asymptomatic, these patients are asymptomatic, but still these patients have direct and high risk contact. So even this patient also have to go for tested for COVID-19. So answer is none of the above, because they asked about except. Which is the main enzyme responsible for the replication of SARS-CoV-2 virus? RNA polymerase is the enzyme which is used for the replication of CoV-2. So that's why we studied about RNA dependent RNA polymerase. What is the most common technique currently being used for viral detection in COVID-19? Yes, the most gold standard test is RT-PCR test. Antibody testing in COVID-19 which detects both IgG and IgM. So answer is both A and B. Full form of RT-PCR, very easy question, right? Reverse transcriptase polymerase chain reaction. RT-PCR which detects which RNA? mRNA is the answer. And confirmation for the diagnosis of COVID-19 is for the screening, we can go for RT-PCR, but the confirmation, the best test is about nucleic acid sequencing. Keep this in mind. What are the viral genes targeted in the diagnosis of COVID-19? There are two types, semi-confirmatory gene and confirmatory gene. So N gene, E gene and RDRP, that is RNA dependent RNA polymerase. All these are gene for the diagnosis of COVID-19. Just radiograph of COVID-19 patients reveals bilateral multilobar radio opacity. You can see the radio opacity present in a bilateral area in case of your COVID-19. And here they asked about what is called as crazy paving. Crazy paving is nothing but lobular septal thickening with various alveolar filing. So this is what called as crazy paving. So this will be discussed during our radiological examination of COVID-19 cases. So which laboratory test has been designated as COVID-19 testing in India? So that is responsible by NIV, National Institute of Virology, Pune. And this is called as nodal laboratory for India. And who is the nodal officer in a district? Collector is the nodal officer in a district. Nodal laboratory is NAV. What is the name of the indigenous ELISA kit developed for the COVID-19 antibody? That is Kavach ELISA. So this will come under a project called as Make in India. And it is developed by NIV, National Institute of Virology, Pune. So these are the various points and concepts which you have to know related to the molecular diagnostic testing of COVID-19. Hope it was very useful. Best wishes for you. Thank you for watching.